Hey guys, welcome to week 8 of PS Parent Leave. I hope you guys have all wished your mothers happy Mother's Day. And yeah, so this week Christian and I are gonna go over two topics, and I'm gonna get started with the first one, which is neurosignaling. And yeah, so I'll be going over neurosignaling, some drugs related, and envenoming of the NMJ, which is the neuromuscular junction. Okay, so I think you guys should know by now the overview of the nervous system, but in case you don't, we're going to go over it again. So the nervous system is divided into the central nervous system and the peripheral nervous system. The central one involves the brain and the spinal cord, and then the peripheral one is it carries messages that's sent from the central one to the rest of the body. So there's the somatic nervous system, which controls your muscles. So these are basically all voluntary. Like you can control like your skeletal muscles, like what you want to do at a given time. And then there's the autonomic nervous system that controls involuntary body functions, such as, I don't know, like digestion. You don't actively think, oh, I want to digest right now. And the autonomic nervous system is further broken down into the sympathetic nervous system and the parasympathetic nervous system. And the sympathetic one is basically your flight or fight response, or is it fight or flight? I don't know. Yeah, whatever it's called. And the other one is for the rest and digest response. Yeah, and it's just a little revision as well. There's always like a presynaptic neuron, which synapses with the postsynaptic cell while at the synapse, and yeah, the action potential always travels in one direction. Yeah, so these are what the parasympathetic and sympathetic responses are in charge of. So like these are just what happens when they're activated. And I think that professors gave us like some mnemonics last year to remember some of these, but honestly I feel like they're really straightforward. Just think to yourself, what am I like, what do I need in my body when I'm trying to fight or flight? And anything else is and the opposite would just be the rest and digest. So obviously, if you see a threat, you want to run away, your heart will beat faster, your bronchioles will dilate so you can breathe more, stuff like that. So yeah, I'm not gonna go over this in too much detail. Yeah, so the most important part of this topic is understanding the neurotransmitters and receptors of all these different systems. So yeah, I kind of summarized what you have to know. So first you have to know the nerve, like what type of nerve it is, and which nervous system it's found in, it's used in. And then there's the neurotransmitters. This is not that important, but we'll go over their synthesis, the storage, the release, their response, and their termination and removal. And lastly, there is 3 drugs I'm going to go over that talks about the pharmacology of just like some drugs that interfere with the neurosignaling of skeletal muscles. So yeah. So by the end of this PSP, I really hope you can understand this diagram. You can basically ignore the last one because even I'm not entirely sure what that is. I guess it's not really relevant now. But so you'll see there's the somatic nervous system and there's the autonomic nervous system. And for each one, we have different neurotransmitters like acetylcholine, norepinephrine, and different types of receptors like nicotinic, muscarinic, blah, 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 blah. We'll be going over all of that, and you'll have to memorize which belongs to which system. So we're going to start off with adrenergic system. So adrenergic nerves, they're nerves that release adrenergic transmitters, and we're going to go over what these neurotransmitters are. And this is only used in the sympathetic nervous system, and therefore it innervates particularly the gut, the heart, the lungs, the blood vessels. These are all things that you can't actively, voluntarily try and control. And as I said, the sympathetic nervous system is responsible for the fight reaction. So when you see a threat, you want to dilate your pupil to let in more light. You want to dilate your bronchial smooth muscle so you can breathe more. You want to increase your heart rate or the force of your heart contractions so there's more blood pumping through your body so you're ready to run. There's vasoconstriction on the skin because you don't really need any blood there. Instead, you want to redirect it to the skeletal muscles, and you're inhibiting your gastrointestinal system because, you're not trying to digest right now. So this is what we're looking at. Yeah. Okay, so adrenergic neurotransmitters are adrenaline, dopamine, stuff like that. I guess the one that we really have to focus on is this one. So yeah, there's subtle differences in the chemical structures between these different neurotransmitters, but because they're mostly the same, I guess, they can interact with similar receptors. So first we're going to look at the synthesis of norepinephrine, or epinephrine. So the precursor is this essential amino acid tyrosine, which is right here, and we don't need to know any of the individual steps, but you just need to know that the rate limiting step is controlled by an enzyme called tyrosine hydroxylase. So therefore, there's a negative feedback loop. So when there is enough of this neurotransmitter that's detected at the cleft of the neuron, this enzyme is turned off by negative feedback loop to inhibit this enzyme. Yeah, and storage and release. So after they're synthesized, they're stored in vesicles, and when an action potential comes, when it's stimulated, it will be released. And something interesting to note is that their storage and release can be stimulated by indirectly acting sympathomimetic drugs. So basically, this kind of means that they mimic the sympathetic nervous system. An example is amphetamine, is that pronounce it? Yeah, and that's like Adderall. So it causes release of these synaptic vesicles, and it has an indirect effect because it doesn't directly act on the receptors, and it is a cause of substance abuse. And then the response generated by these neurotransmitters, we're going to go over in the next page. And lastly is their termination and their removal. So there are two methods in which noradrenaline is removed at this synaptic cleft, and of course this is necessary whenever you want your signal to stop. And the main one is called uptake 1, and so basically your neurotransmitter is taken up again, and then it's broken down by different enzymes into metabolites. So some examples are MAO, COMT, and so they're inactivated by reuptake and then enzymatic breakdown to be reused. And so I guess something special about this is that these neurotransmitters are reused. So I guess when they're broken down, these metabolites can be used to synthesize them again. Yeah, and there's also apparently some abuse potential for this as well. So cocaine inhibits these enzymes, so therefore these neurotransmitters are not removed, and then there's in general overactivation of the receptors, and it causes a lot of stimulation in the nerve. And the other method is called uptake 2, and it's extraneuronal, meaning it doesn't happen in the original neuron but in the next one, so it's taken up by the postsynaptic cell. Yeah, and apparently noradrenaline, dopamine, and 5-HT all have similar uptake mechanisms, thus drugs that affect their uptakes can have similar effects on all of them. And now we're going to look at their receptors, which is responsible for mediating the responses of these neurotransmitters. So for adrenergic ones, the subtypes are alpha and beta. So when noradrenaline binds, a chain of events occur inside the cell, and these are mediated by secondary messengers such as cyclic AMP, stuff like that. And the response, like I said, they're sympathetic responses, and these this diagram gives you examples of, I'm sorry, it wasn't in English, but I was trying to find the most visual diagram as I could, and you'll see what type of receptor subtypes are found in different parts of the body. For example, in the lungs is beta 2, in the heart is beta 1, stuff like that. It's not that important to remember this at this current moment, but you'll have more you have more lectures coming up where they go over this again, and eventually you'll see it's quite important to remember some of these. Yeah, so for example, in the heart, it's mostly beta receptors, and the response is to increase the rate and force of contraction. So therefore, drugs targeting this, drugs trying to target hypertension, so antihypertensives, are beta blockers, so they're antagonists to lower the blood pressure, right? They're trying to, because they're antagonists, they're trying to do the opposite of this effect, and that's how you lower hypertension. And for example, in the blood vessels, there are both alpha and beta receptors found, and they're in charge of increasing vasoconstriction, and vasoconstriction always leads to increased blood pressure. So in addition to antihypertensives that target the heart, there are also ones that target specifically blood vessels based on the type of receptor subtypes that they target. And then lungs, it's mainly beta receptors found there, and it's in charge of bronchodilation. So of course, if you have asthma, you try and relieve it. We have beta agonists, so we try and to amplify this effect to relax the bronchial smooth muscle and allow the patient to breathe more easily again. And in the gut, it's both alpha and beta receptors found. So not only are they alpha and beta, I guess they're like further divided down into like alpha 1, alpha 2, beta 1, beta 2, but yeah. It is not that important in this like immediate moment. And now we're going to move on to cholinergic nerves. So cholinergic nerves are found both in the parasympathetic and somatic nervous system. So here is the somatic one, you see, and there is also the parasympathetic right here. But something else that's important to note about cholinergic nerves is that they're always the presynaptic neuron. So the presynaptic neuron, whereas synapses with the postsynaptic one, it's always cholinergic neurotransmitters and receptors. That's one thing to remember. So it's in charge of, once again I'll say it's in charge of somatic and parasympathetic and the ganglion in between every time. So it innervates, since it's somatic, innervates voluntary skeletal muscles and involuntary ones, which is the heart, the gut, and your blood vessels. These are just stuff you can't control. So cholinergic neurotransmitter is acetylcholine, and the synthesis, once again, we don't have to know this in detail, but luckily this one's easy anyway. It just choline is converted into acetylcholine using this enzyme called choline acetyltransferase. And storage and release. So they're also packaged in vesicles, and when an action potential comes down, they're brought to the cleft and then exocytosed. And response, once again, we're going to go over that on the next page. And their removal and termination is quite different from adrenergic neurotransmitters. So it's mainly via enzymatic breakdown by this enzyme called acetylcholinesterase. I hope I pronounced that right. And it is not reused. And so for acetylcholine to function at most neuromuscular junction, it must be removed and activated almost immediately. I mean, you can imagine that your muscle movements are quite precise and stuff. So you want the contraction to happen really precisely. As soon as it happens, you want your neurotransmitter removed. And this is much faster than muscarinic cholinergic, which we'll see more in the next step. And lastly, this enzyme, this acetylcholinesterase, is very abundant at the synapses. You'll find a lot of it at every synapse of neuromuscular junctions. And now this is the responses. So cholinergic receptors are nicotinic ones or muscarinic ones, right? And nicotinic is the one that's found in the somatic nervous system, so in charge of the muscles, while muscarinic is the one that's used in the parasympathetic nervous system. And I remember I was always so frustrated last year. I was like, surely muscarinic ones are supposed to control the muscles, but so if it helps you, you can think that it's the opposite. The muscarinic one doesn't control the muscles, while the nicotinic one does. So the reason they're called nicotinic is because apparently they're activated by something called nicotine. And these receptors are gated ion channels that open when acetylcholine binds to the binding site. And of course, what happens is in the muscles, it just causes contraction, or you can also find it in the ganglia in the brain. So that means when it says ganglia, it just means what I was talking about here, like it's found in ganglions. So you'll see they're all nicotinic. And while in a PNS, it's a muscarinic one. So once again, this is parasympathetic, it's muscarinic receptors. Muscarinic ones, PNS, it's called muscarinic because it's activated by muscarine, which is a natural compound apparently formed in poisonous mushrooms. So it's responsible for the PNS, which is involuntary, rest and digest, and basically if it helps you remember, is anything that's the opposite of the alpha, beta, adrenergic, sympathetic response. So yeah, some examples are in skeletal muscle, which, like I said, was nicotinic. It causes contraction, but in the autonomic side, in the heart, it would decrease the rate and force of contraction because you're resting, you're digesting, you don't need your heart to beat fast. And it promotes contraction, it promotes peristalsis, it promotes the digestion of your food. And what's responsible is the muscarinic receptors, and apparently blood vessels are not innervated by these receptors. And cholinergic effects, there's a few examples over there. Oops. And lastly, I'm going to go on to neuromuscular blocking drugs. So drugs can interfere with the synaptic transmission in two ways. One is presynaptically, so we do it by inhibiting the acetylcholine synthesis and release. So yeah, anything before the thing gets released, or it can happen postsynaptically. So basically, the acetylcholine is already released, but you're preventing it from binding to the receptor, or you just inactivate the receptors in general. So like inactivate the channels. Yep. So there are two kinds. One is non-depolarizing blockers, and the other kind is depolarizing. So non-depolarizing means the muscle doesn't get depolarized at all. It doesn't get stimulated at all. While depolarizing means it actually does, but somehow it's still a blocker, which we'll see later. So first, so non-depolarizing blockers are competitive antagonists at N receptors. So basically, they compete with acetylcholine to bind to these nicotinic receptors. So when they bind to it, the acetylcholine can no longer bind to it anymore, and these ion channels can't open, and the motor end plate can't depolarize. And so then it makes sense that the action of it can be overcome by increasing the concentration of acetylcholine, because the more acetylcholine you have, the more it's fighting against this drug. And a type of drug that can do it is anticholinesterases. So they're anti against the enzyme that breaks down acetylcholine. So since acetylcholine is not being broken down anymore, you have an increased concentration of acetylcholine. And an example of this drug, which you're supposed to know, it's an E-pharm, is pyridostigmine. I'm really hoping I'm pronouncing that right. So yeah, so there's no initial stimulant action at all. So they don't turn on the receptors on at all. They just block them. And apparently they're not absorbed orally, as it's highly polar, and instead they're given intravenously, and they don't pass the blood-brain barrier or the placenta. But yeah, and all of these drugs, these three drugs that I mentioned today, I would recommend you going on ePharm, just to give it a read yourself. I think it wasn't that important to remember all the details on ePharm, but I think it's worth it. Just take a look and see if you can find anything interesting. And so some examples of these drugs are tubocurarine. Oh my god. Pretend I pronounced that right, okay? And so the interesting thing is that this drug is not actually found on ePharm, but it actually showed up on our exams last year and stumped a lot of people. So yeah, I would recommend you to know it. And the best way to memorize it is that this is a compound that's used by many tribes to hunt animals, and they paralyze them. And the reason they do it is because since this drug is not absorbed orally, it doesn't actually hurt the animals, it doesn't hurt their meat, so you can still eat them safely. And they have a pretty long duration, 1 to 2 hours, so therefore they're really not used that much anymore in the clinical context nowadays, as there are many more short-acting ones. And the side effect of this drug is ganglion blockade and histamine release from mast cells. So it can also cause all these like bronchospasm, rash stuff type of symptoms. Yeah, and so what is used more nowadays is synthetic derivatives, which have much less side effects and shorter times of onset. And there they are, and the one that you have to remember is rocuronium. Why does I have to do farm? Rocuronium. Yeah, and I remember the seniors last year try to memorize this basically by thinking, oh, it's like a rock, so you can't move. It's like a rocuronium. Yep. And now we're going to move on to depolarizing blockers. So somehow they stop your muscles from moving, but they still depolarize, it feels counterintuitive, but it makes sense. So they're nicotinic agonists, and receptors. So and receptors are what's found in skeletal muscle, and basically overstimulate. They stimulate it so much that the motor end plate becomes unresponsive and it can't repolarize again. And if it can't repolarize first, it can't depolarize again. So yeah, so even though they're agonists, they're actually blockers, and they have similar action to acetylcholine. So of course the action is further increased if there's an increased concentration of acetylcholine. Yeah, so they act on muscle end plates to prevent cycles of depolarization and repolarization needed to sustain a muscle contraction. Yeah, so they basically work by inactivating the sodium channels. Sodium can flow in. The receptors are also unresponsive to acetylcholine. And the example for it that you have to know is succinylcholine, which acts for 3 to 7 minutes, and it's basically 2 acetylcholines stayed together. So that's 1 acetylcholine, and this drug is 2 of them. Yeah. This week is Envenoming, and this was an active learning session last year, and I just want to let you guys know that this content is very low yield. I actually had no notes from the session, neither did anybody in my PSP, and this content never ever came up in any of the exams. So yeah, I'm gonna direct you to this source, which can be found on the Mnemonic academic drive. So if you have time, go check it out. But honestly, I think it goes into way too much detail, and it really isn't anything important. So I'm just gonna try and go over some of the stuff that I personally think may be more important. Okay, and so the first one is the difference in terminology between venomous and poisonous. So venomous is any creature that actually has a specialized apparatus to inject the toxin into your body. So it could be glands or specialized cells, ducts, or stings, fangs, spine. And poisonous just means that the creature doesn't have this special apparatus. Instead, the toxin must be absorbed or inhaled or ingested. So yeah, it must be touched or ingested. Yeah, so the different types of toxins, there are neurotoxins. Neurotoxins target neuromuscular junctions. So you can see 2 types here. So they can either be presynaptic, and that just means they're affecting the neurotransmitter, like a synthesis or something like that, or its release, or it could be postsynaptic. So it's targeting the receptors. And there are also hemotoxins. So hemo means like blood, so they make your blood coagulate. And lastly is myotoxins. So myo means muscles, so they target your muscles, and here they are. So this is your neuron, the terminal, and then it's where it synapses with your skeletal muscle. So you'll see by the diamonds here that the toxin is targeting the muscle tissue. And LD50 is the dose of toxic agent that is required in order to kill 50% of a population within a given time frame. And lastly is this treatment. So for some of these things, the first step in treatment is pressure immobilization. And I think a lot of you probably have already done the first aid course that the faculty recommended, and you would know that it's basically trying to like bind your limb or whatever got bitten to like a stick or anything solid to try and get it not to move as much as possible before you get medical attention. And the purpose of this is to restrict lymphatic flow. So every time you move, the toxin gets pushed around your body a little bit more and spreads to more parts of your body. So by immobilizing it, or basically trying to like bind it off, you prevent it from getting into the circulation. And the next thing is antivenoms. So they can be monovalent or polyvalent, meaning whether they're effective against only one kind of venom or many types. And I think the biggest takeaway from the active learning, which these are points that Christian mentioned, is that their uses are actually a lot more limited than we think. So first of all, when we get stung or whatever, we may not actually know what actually hurt us, so we wouldn't actually necessarily know which antivenom to use. Next, most of the time when people get bitten or stung or anything, they're in the wilderness, far from any hospitals, any medical care. So by the time they actually get some, it may be too late to use the antivenom. And lastly, a lot of them are just not as good as we thought. So I remember watching several videos about it during the session, saying that all these standard protocols that used to be used, when they actually researched into it, they found that it actually didn't improve outcome at all a lot of the times. So yeah, so that's it for a topic. If you have time, go check out this. It has a lot more detail on some other things.
Okay, so welcome back to another session of PS Pain Relief. I know that it's kind of like week 8, and week 8 for me is roughly the time I start to feel like the semester has been long. The semester has dragged out for quite a bit. I don't know if you guys are feeling the same thing, but if you are, it's completely normal, and if you're not, then you're um, an alien. But you know what? We're all here to learn. We're all also here just to be, just to help you guys. So if you guys have any questions, any concerns, please once again direct it to us via DM. We are going to be happy to reply to your DMs. And yeah, just don't worry too much about this kind of end of the semester burnout feeling. It's completely normal, and just make sure that you talk to your friends. And with that, let's get on to the next slide. Okay. So what are we going to cover today? We're going to be covering the 4 tissues that form organs. So we're going to put in some histology. Honestly, once you know, you kind of know. We're also going to be covering the integumentary system. That is also not super, not super major. You guys should just know the histology of everything. So that part's going to be covered mostly by my friend Christian, so it will not be me. And I'm so sorry. My energy is really off because I need a nap and it's very cold. Okay, so the 4 primary tissue types, what are they? They're the nervous tissue, connective tissue, muscle, and epithelial tissue. So all you guys really need to know here is the different appearances that these tissues take on when they're observed under a microscope. So this, as you can see, it's nervous tissue. You can see these would be the cell bodies, and these would be the either the axon or dendrite terminals. I am not good at this, but we are all here to learn. So these these are either the axon or dendron terminals, and nervous tissue makes up the brain, spinal cord, and nerves. Connective tissue is bone, fat, or soft tissue, and tendons. And here you can see the very irregular appearance, very kind of like loose connective tissue. And next up we have muscle. We have cardiac muscle, skeletal muscle, and smooth muscle. You guys would have learned about these types of muscle earlier. And as you can see, well, it's not very obvious here, but if you have the slides and you can zoom in, you can kind of see that there are like these mini like really small striations, and again you can see they're arranged in fibers. So this is just the typical appearance of skeletal muscle. And also for epithelial tissue, this is what we're going to be covering today in more detail. These form the organ linings. So as you can see, the lumen is like the inside of the pipe of whatever pipe is inside your body. I know that sounds gross, but come on, it's week 8. Okay, and then you have stratified squamous epithelium. You have a basal layer. So just remember that every form of like epithelial tissue consists of the basal layer with cells on top of them. And just remember that in cancer, we know that it's really bad when a cancer cell is able to kind of infiltrate the basal layer, because as you can see, the basal layer is really defined, and none of these cells are actually able to infiltrate and just kind of burrow deep into the basal layer. So you know that it's bad, and you know that it's definitely abnormal and characteristic of cancer when it's able to infiltrate the basal layer. So anyway, enough of that. You guys would kind of notice at this point that you guys have basically covered the details, the structure and functions of all of these tissues already. This is just really, this should just be revision at this point. And also, you know, if it's not revision, then at least you guys are learning about histology from this. So yeah, don't worry about this really. Okay. The integumentary system. What is it? So basically the integumentary system is the combination of skin and its like associated appendages, okay? So it consists of the epidermis and the hypodermis. And its functions are to protect, if you remember, about your immune system. The innate immune system has the skin as a barrier, it's waterproof, and it protects us from external pathogens. Unless we kind of have a scrape or have a laceration, then in that case, yeah, infection city over here. Absorption, if you guys have ever seen those those medicine patches that you can kind of stick on yourself, or I am forgetting what they're called, mostly because I'm tired. But yeah, there are some medicines that you can absorb via your skin. When you moisturize your arms, you also can feel that the skin is absorbing the like moisture. And excretion. So remembering that a lot of the ammonia in our bodies, a lot of the excess like salts and stuff are excreted through the sweat that we produce. So that's excretion. Secretion, remembering that our skin also in certain areas produces antimicrobial compounds to fight off bacteria and other forms of pathogens. Regulation would mostly be with temperature. Um, so remembering how when it's cold, you have your erector pili, which make your hair stand on end. Also your skin has it has small capillaries that can undergo vasodilation or vasoconstriction depending on how hot or cold it is. And also there is sensation. So remember that your skin contains, actually not remember, sorry, we're going through that in this lecture. But remember that your skin has nerve endings, sensory receptors that are really useful in helping you communicate with the outside world. And just a short note that this is not how you should be using your integumentary system or the integumentary system of other people. So bear that in mind. So what are the components of the integumentary system? And I know that this is a lot of text. I put the diagrams on like the next slide. But anyway, basically the integumentary system consists of epidermis, which are stratified squamous epithelial cells, and the dermis, which are dense irregular connective tissue. Okay, so you can see that the integumentary system, even though we interpret this as being like skin, it is not strictly epithelial tissue. There's also connective tissue in it. So anyway, we have the stratum corneum, which is the most superficial, and the stratum lucidum. Both of these have dead cells and keratin, except that the stratum lucidum is only in thick skin. We also have the stratum granulosum, which have the lamellar bodies, the stratum spinosum, which have keratocytes, which, if you can kind of guess by the name, they produce keratin. And we have the basal layer, which is the deepest layer of the epidermis. And as you recall from me saying just now, epithelial cells are always situated, always kind of sitting on top of a basal layer or a basal lamina. And these are the cells that will, and these are the cells that will multiply and slowly advance up the levels. So for the dermis, on the other hand, we have just the papillary layer, which is the most superficial layer. It's got loose connective tissue, some nerves and blood vessels, and it's also got the reticular layer, which has more collagen and it's less elastic than the papillary layer. Okay, and then next slide. We just have some mnemonics here. Well, okay, I can't move my picture thing, but you can kind of guess that it says deep to superficial. So, oh, this is so funny. Okay, so we have the stratum basale, spinosum, granulosum, lucidum, and corneum. So that's going from deep to superficial. So from deep to superficial, it's British, Spanish grannies love cornflakes. Okay, you can tell that I didn't make the slide, but you know what? I like cornflakes. Everyone likes cornflakes. And so do British and Spanish grannies. So that's just a way for you to remember the different layers of the epidermis. And now Just some pictures. You can see again, British and Spanish. Grannies love cornflakes. There you go. From deep to superficial, and the dermis. You can see this is the papillary layer. It's kind of lighter. It's got the ends of the nerves and it's got the ends of the blood vessels and the anastomosis right here. And the reticular layer is deeper, and it's got more connective tissue. As you can see here, once again, there is a distinct difference between the two. Remember that I think papilla means like, doesn't mean butterfly or something, but it means basically like wings or something like that. So you can see these folds here. They kind of look like butterfly wings from the side. Okay, and that is about it for me. I'm going to pass the time on to my friend Christian, who's going to tell you guys about the different components of the integumentary system. See ya.
Hi guys, I'm going to be taking you through the rest of the integumentary system, picking up where Mayanne left off. So you guys would have been given this table probably during your active learning session, or at least in the slides. Now, a lot of this table I would say is fairly low yield stuff / completely unimportant. So I'll skip over most of it. For instance, do you really need to know that sweat glands found in the anogenital region resemble mammary glands? Probably not. They may have important clinical relevance, but not to you guys. What is important is basically the stuff on the left and understanding more or less the general function. So don't worry about the stuff in blue, green, and red, just the stuff in white here and understanding a global idea of how they work. So without further ado, let's get straight into it. So to start with, sorry. Let's go back. So to start with, the epidermis of thick skin. Mayanne was probably talking to you about the layers of thick skin and how they compare in the layers of thin skin, particularly in the epidermis. And you may remember that thin skin, being thinner, actually lacks one of the layers of the epidermis. And that layer, see if you can remember what it is, you can pause the video, is the stratum lucidum. So the stratum lucidum is missing from the epidermis of the thin skin. There's only 4 layers of epidermis. We don't need to know the exact figures of the difference in size, just that the epidermis of thick skin is a lot thicker, and it's primarily due to a much thicker stratum corneum, which is that very thick keratinized layer located superficially. Melanocytes, these release a dark pigment called melanin, which is what gives our skin their complexion. So darker skin has more melanin and probably therefore more, or at least more active, melanocytes. Now these guys are found in the stratum basale of the epidermis, so quite deep in the epidermis, but not in the dermis. They're in the epidermis. And they release their melanin and transfer it to the cells above in the epidermis, and it protects their DNA. Then we've got our dermis. This is a layer below the epidermis, which itself is comprised of 2 sublayers. It's primarily dense irregular connective tissue, but it's got a superficial layer, which is perhaps the more important layer. It's the papillary layer of the dermis, and this houses a lot of loose connective tissue as well. You will see that in slides shortly. We've got sweat glands, so they produce sweat. There's 2 kinds not listed in this table. We've got apocrine and ecrine. May not be tested on this, but apocrine is found in hairy areas. So for instance, in your armpits, on the top of your head, and in your genital areas. And basically they're associated with hair follicles and they produce really smelly sweat. So they originated from older sweat glands that were designed to release basically pheromones, so that we could kind of communicate by smell as well. But the need for humans to do that has obviously lessened, and now in fact we usually find the pheromones from our armpits to be quite repulsive. So now we shower and we really resent our apocrine sweat glands. Ecrine sweat glands are found all over your body and they help to keep us cool. They release a more watery sweat. And we've got sebaceous glands. These guys are also associated with hair. They're located near the roots of our hairs and they release sebum into those roots. And then as the epidermis starts to cycle through and as the hair grows, this pushes that sebum slowly out and more superficial. And it's designed to basically provide a fluid that traps bacteria and stops them from getting into our dermis. Myoepithelial cells. not massively important, but it's important to understand that there is a little bit of smooth muscle that surrounds our glands in our skin, and it squeezes particularly the sweat out of our skin. So that's what the myoepithelial cells do, very important. Then we've got our Meissner's corpuscles and our Pacinian corpuscles. The faculty seems to love these for some strange reason. Apparently, they're kind of important. Basically, they're sensory nerve endings, and they're shaped a little bit differently. They're located a little bit differently. I've got a mnemonic for how to remember this momentarily. So let's have a look at this histology here, okay? So what I want you to do is pause the video and see if you can identify the I've given it away, the 5 layers of the epidermis of thick skin, all right? See if you can remember what they are, and I'll give you my version of what I think everything is. Okay, so here we go. I think that this superficial pink layer here, probably down to about here or so, or maybe a little bit deeper, would be what we call the stratum corneum. So I'll put SC. And this is primarily that very flattened, mostly dead squamous skin cells, right? So on thick skin, we've got a very thick stratum corneum. So lots and lots of dead skin. And basically this will slowly flake off and abrade the more stuff we do with our hands. So it protects the living cells that are underneath. And obviously on thick skin, which is high traffic areas like our hands and the soles of our feet, we need more of this stuff. So that's that very pink layer. Now underneath that, we have what we call the stratum lucidum, which is unique to thick skin. It's called that because supposedly it appears a little bit lighter on histology slides. Now it's not very evident here, but it would be existing somewhere around here, this stratum lucidum. So I'll put SL for stratum lucidum. Now, then after that, we have a very thin layer underneath the stratum lucidum called the stratum granulosum. And you can kind of see the cells of that here. They're a little bit darker in color. You can see some dark nuclei. That's kind of where your stratum granulosum is. So I'll put little SG there. Nice. Then we've got a purpley layer underneath. And you can see it has little spine-like projections that go into this lighter area, which is called the dermis. But this purple area, which is fairly thick, is called the stratum spinosum. So we'll put SP. And that extends from here to about here. And then finally, we have a very thin layer again, just a couple cells thick here. And it goes all the way around our stratum spinosum. And this is called our stratum basale. So this is what houses our melanocytes. All right, here we go. Let's have a look at the epidermis of thin skin. Remembering that this has 1 layer less than our epidermis of thick skin. See if you can remember what layer that is. It is the stratum lucidum. Did you remember right? If you did, well done to you. If not, no worries. It's largely a useless piece of trivia. All right, onwards. Epidermis of thin skin. We can see a little bit of stratum corneum. It's that very densely pink colored section, which has kind of flat, flaky bits. Underneath, we've got a fairly thick purple layer with projections that go down into the dermis. This is probably the stratum spinosum due to those spiny projections. We can't see the stratum granulosum too well, but it's probably those dark nuclei that we can see between the pink and purple layers here. You can see, like, for instance, maybe about here there's a little dark nucleus, and maybe there's a little dark nucleus. Well, those are probably nuclei of the stratum granulosum. And then underneath, not very visible to us, we have the stratum basale, which runs all the way along this kind of purple area. And it's 1 to 2 cells thick, maybe. And it's basically attached to a little basement membrane down below. And remember, it has melanocytes, so it's what gives our skin its pigment. Okay, we can also see dermis in here. We've got this fairly loose connective area with little projections upwards. So those projections upwards, which I'm drawing lines in, are what we call the papilla. So we call this loose connective area the papillary dermis. Underneath, we've got a more dense layer, which kind of has a net-like structure around here. This is what we call the reticular dermis. Melanocytes here, we've got a little closer view. They're these darker colored cells in the stratum basale here. And we can kind of see in this area, we've got stratum spinosum, and this lighter colored area would be dermis. You can see more dermis here, these light areas. Now remember, the papillary dermis is loose connective tissue, which means that the cells are far apart, and most of this area would be composed of ground substance or extracellular matrix, essentially. So as a result, this stuff, this extracellular matrix, doesn't take too well to the stain that we use in histology slides, so it appears lighter. So that's how we can remember that. And down here, it's denser, so the cells are a bit closer together, and that's why we can see it. Sweat glands, not massively important. Let's skip it. Sebaceous glands. Well, here we can kind of see a cluster of, perhaps it could even be 3. I don't really know. But you can see the sebaceous glands here at the base of the root of this hair. Now, the follicle of the hair is actually deeper, okay? So we've got the root of the hair, which is this epidermal part, but down below is a dermal part, way below these sebaceous glands, probably a good 2 inches below this slide. Now, what the sebaceous glands do is they've got little ducts, you can actually see it here, that connect them to the base of the root of the hair here. And those ducts deliver the sebum into this area, so it pools with sebum. And then the hair grows and the skin cells cycle outwards, pushing this sebum out into the outside world. And the sebum collects bacteria as it goes, stopping them from getting into the dermis, because this is essentially, because it intrudes into the dermis a bit, it's a weak point, and bacteria would love to get in there. That's why we have sebaceous glands associated with our hair roots. All right, on to the next thing. All right, more histology. Let me just clear some stuff. Okay, so myoepithelial cells, not massively important. Let's talk about Pacinian corpuscles. They're very easy to identify. They've got this sort of layery structure here, look a bit like onions. Mayan's kindly included a mnemonic meme for us. Pacinian corpuscles are like onions, they have layers. Now, Pacinian corpuscles are located quite deep in the dermis, almost near the papilla of the, almost near the hypodermis. All right, then we've got our Meissner's corpuscles here, and these guys are located quite superficially, okay? So you can actually see here, this is epidermis, this dark purple layer, and the lighter area is more like dermis here. So we're talking stratum spinosum, this purple area, and probably papillary dermis in this area here of the Meissner corpuscle. Hard to spot, it's this little interesting arrangement of cells that you can see here, slightly unusual. Now, my mnemonic for this is, I primarily use the first one, but the second one's kind of an accessory mnemonic just for completion. So I remember that Pacinian corpuscles, P for pressure. So they detect pressure, first of all, and sustained pressure, deep pressure. Those are the main types of pressure. Now we can remember also that they must be found deep in the dermis, because to detect deep touch, they have to be deep in the skin, which makes sense. But another way you can remember it is the second line of the song, Under Pressure, which is under pressure, pushing down on me. So because it's down, deep in the skin. I also remember that they're responsible for detecting vibration in part, because Under Pressure is a song gives you music and sound gives you vibrations. Then finally, I remember the Meissner's corpuscles. I remember meticulous, Meissner, meticulous, and meticulous touch would be fine touch. So they have to be located more superficially in the skin to detect that type of touch. Again, not really massively clinically important, but for some reason the faculty thinks it is. So onward to the next thing. And this is just a small aside. You guys may not have done injections this year. I hope you did, because it was really fun to do when we did it. But obviously COVID has really thrown a spanner in the works. If you did, you would have learned about subcutaneous and intramuscular injections. So we're going to talk about subcutaneous injections. They go into an area of skin called the hypodermis, which is just underneath the dermis here. And it's primarily fatty adipose tissue, so loose connective tissue. It's very, very vascular, lots of arteries and veins and stuff run through there. And that's really good if you want to put, if you want to inject a medication or a vaccine or something into that area, because it means it's going to very readily be absorbed into the bloodstream. So subcutaneous injections, their target is this hypodermis here. Now, intramuscular injections, the injection goes a little bit more perpendicular to the skin, and it goes down into this muscle layer just below. So that's the primary difference. Anyway, I hope that was helpful for you guys, and happy studying. I wish you the best of luck.